In our final lesson on techniques used in the study of proteins, let's consider together the topic of proteomics. Just as there is a genome representing the genetic content of the cell, so there is a proteome that represents the total protein content of the cell. Proteomics is therefore the study of the content and interactions of cellular proteins. We might approach this study in such a way as to determine the structure of the proteins produced under a given set of parameters. We might instead be interested in the total protein content of the cell, that is to say, which proteins are expressed under varied circumstances. Lastly, rather than total protein content, we might be more interested in specific protein interactions. Let's consider a method sometimes used in this last type of proteomics study. In this example, we are studying a target or bait protein that has been genetically engineered so that it carries a molecular or affinity tag on its surface, as illustrated by step 1 of our figure. The presence of this tag allows us to isolate the protein using affinity chromatography. We allow our bait protein to interact with cellular proteins under a given set of conditions, and these proteins are illustrated here by the shapes numbered 2 through 5. In step 2, a cellular extract is applied to the affinity column and the bait binds via the surface tag. The bait is then eluted from the column in complex with the other proteins. In step 3, the proteins are isolated using SDS page and the protein bands are excised from the gel. In step 4, each protein can then be subjected to analysis by peptide digestion, followed by mass spectrometry, to determine the primary structure or amino acid sequence. Finally, bioinformatics techniques allow us to identify each of the proteins involved. We will consider many examples of protein interactions in our study of biochemistry, and this is one example of the techniques employed in making those determinations.